Bismillahirrahmanirrahim. Welcome again, guys. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about termination or elongation of DNA transcription in eukaryotes. Okay, in my previous video lecture, if you guys remember, we talked about the proteins, we talked about enzymes, we talked about transcriptional factor, and we also talked about the overview of the DNA transcription in eukaryotes. And we also talked the initiation of DNA transcription as you watched. Okay, if you missed these videos lecture, it is present into my channel, go back and watch it. Today in this video lecture we are going to talk about another stage of DNA transcription. We call it elongation of DNA transcription. So how the elongation of DNA transcription will be carried on, and what are the main requirements in elongation of DNA transcription, we'll be talking in this video lecture. So please stay tuned and continue to watch. So let me erase the portion and let me start. Okay, so let me write. Okay, now what happened in this case? To explain the process or the step involved in elongation of DNA transcription, first of all, let's talk the introduction of elongation of DNA transcription. You know, elongation of DNA transcription in case of eukaryotes as well as in case of prokaryotes is similar with each other. So if you want to understand the elongation of DNA transcription in eukaryotes, you must watch the prokaryotic video lecture, elongation of DNA transcription in prokaryotes. Once you get idea about that, then this one video will be very easy because both are similar to each other. But a little bit transcriptional elongation factor are involved in this case, due to which it will make the elongation of DNA transcription a little bit complicated. Okay, so first of all we need to talk to the requirements. Now first of all, let me tell you what is the elongation of DNA transcription. Again, elongation of DNA transcription is the stage in DNA transcription in which the number of nucleotides are added one and after another by RNA polymerase, and it will make a long chain of RNA by using the DNA strand as a template, which have 5 to 3 prime which have 3 to 5 prime end, and after that we get a long chain of RNA. This is called elongation of DNA transcription. Okay, now what kind of requirements we need in elongation of DNA transcription? So there are different kinds of requirements. So let me write requirements. So what we need in this case? In this case we need RNA nucleotides, which are mainly found inside the nucleus, because as we are talking the DNA transcription in eukaryotes, so definitely nucleotides will be found in the nucleus. Now in case of prokaryotes, it will be found in the cytosol of the cell. We need RNA polymerase 2. So inshallah in upcoming videos lecture we are going to talk about RNA polymerase 1, the transcription of transcription done by RNA polymerase 1 and transcription by RNA polymerase 3. But here I make the videos lecture only by RNA polymerase 2, transcription of RNA polymerase transcription by RNA polymerase 2, and how it will be carried on. So it is very important because it can synthesize, if you guys remember we talked that it can synthesize all the necessary RNA, means messenger RNA, transfer RNA, ribosomal RNA, miRNA, snRNA, hnRNA, a lot of RNAs. This one RNA polymerase can synthesize, have the ability. Now moreover we need a protein bridge. So if you guys remember, we also talked that protein bridge is also need in case of prokaryotes as well, but in this case we also need the protein bridge, and with this protein bridge we also need some other elongation transcriptional factor 2. So there are many of the elongation transcriptional factor 2. Okay, now what are the main function of this elongation transcriptional factor 2? The main function of this elongation transcriptional factor 2, it is involved to start or to enhance the transcription process. We can say enhance. It involves in enhancement of transcription. Okay, and it can also involve to slow down the transcription process. Slow down. And we can also say that it is also there are different kind of elongation transcriptional factor 2 which are involved to stop the transcription as well. So it also involved in the stopping of DNA transcription. So it means that this elongation transcriptional factor is very important. Elongation transcriptional factor 2 is very important because due to this elongation transcriptional factor we can either enhance the transcriptional process. So there are some sequences. This elongation factor encounters some kinds of sequences which are found in the DNA. Due to these sequences, the elongation will be fast, will be carry fast, and it will after that it will found some of the or it will encounter some of the nucleotide which are found in the DNA, and due to these nucleotide or sequences, the transcription will be slowed down. And after some time it will encounter some other nucleotide, and on that nucleotide it will be stopped, and after that the termination of DNA transcription will be occur. So we will talk it later. Now there are many of the elongation transcriptional factor we have, but the most important elongation transcriptional factor are P, T, E, F, B. Okay, so transcription elongation factor B. So we need this elongation transcriptional factor 2, which are very important. Now what are the main job of this elongation transcriptional factor 2? It is mainly involved in addition of RNA nucleotide. So it is involved in addition of RNA nucleotides, and the second function is that it also activate and phosphorylate spt5 and TATSUF. So let me write activate and phosphorylate late spt5 and TATSUF. Okay guys, now remember, among them this TATSUF is the universal transcriptional factor. And why it is called universal transcriptional factor? Because it is involved in the 5' capping, and moreover it is involved in the CDT phosphorylation. So that's why we can say that it is the universal transcriptional factor. And at the end we need another elongation transcriptional factor. We call that elongation transcription factor 2S. So let me write here. What are the main job of this elongation transcription 2F? It mainly involve, or it can perform the exonuclease activity. Exonuclease activity. Okay, so these are the main things that we need in elongation of DNA transcription. These are the requirements. So if you guys remember, we talked that these three main things which are mainly need in prokaryotes, but in eukaryotes some other requirements we also need, due to which it can make the eukaryotic DNA transcription a little bit complicated. Now let's talk about the process or the steps which are involved in this case. Now what happened, again we have a DNA molecule, as you can see. Okay, so let's suppose this is a DNA molecule having one strand having 5 to 3 prime end, while the other strand have 3 to 5 prime end. Okay, and we have also the RNA polymerase here. So let me draw. So let's suppose this is the RNA polymerase, mainly RNA polymerase 2 enzyme, and it have also the transcriptional factor 2H, transcriptional factor 2H they have also, and they have also the C-terminal tail, as we talked in elongation of DNA transcription. So what happened? This RNA polymerase 2 have the ability to add nucleotide one in after other. If you guys remember, we talked that the DNA are bended in the, but whenever now once the elongation of transcription start, the DNA will be linear in form, and again it will come its in original form. So now DNA is not bended more, and it is now in its original form. After that, what happen? RNA will be transcribing. So this is the RNA which are transcribing, as you can see. But here one question arises in that how it is transcribing. It mainly transcribes that DNA RNA nucleotide will be added one and after the, and all the process will be carried on inside the nucleus. Why? Because this is the DNA transcription in case of eukaryotes. So it is going on inside the nucleus. And what happened? Let's assume this one portion, so we will easily understand the elongation of DNA transcription. So let's suppose this is the lower strand or upper strand, or any strand having 5 prime to 3 prime to 5 prime end. So let's say, okay, so they have 3 to 5 prime end. So these are the nucleotide DNA sequences, as you can see. Okay, and some of the sequences are flipped downward. Okay, now what happened in this case? This is, let's suppose, this RNA polymerase 2, and this is the strand which is synthesized and elongated. So remember, this transcriptional bubble should be needed, means the DNA should be melted one and after other. Once the DNA are melted, when the DNA strand are separated or transcriptional bubbles are formed, then the RNA polymerase will be moving. Okay, this is very important concept. So RNA polymerase 2 have no more ability to move, but it will be moved when the transcriptional bubble is formed, when the open complex is formed. After that, the RNA polymerase will be moved in left. According to me, it will be moved in the left direction or in the forward direction. Then what happened? Here the protein bridge comes. So let's suppose this blue one can represent the protein bridge. Protein bridge. Now what are the main function of this protein bridge? If you guys remember, we talked in my previous video lecture. The protein bridge will flip one of the nucleotide in upward direction. So it will flip this nucleotide in upward direction, as you can see, and then it will move in the forward direction and then flip another nucleotide in upward direction. So it can flip one of the nucleotide at a time. It don't have the ability to flip a lot of nucleotide in one time. Now it will be flip the nucleotide in upward direction at one time. So once it flip the nucleotide in upward direction, then it will be carry on, and after that the RNA polymerase will be also carry on, and means they can follow this protein bridge. So whenever it is moving on, so then they will detect the nucleotide, and if they detect that if there is guanine, let's suppose they detect that there is guanine, so what actually they will bring cytosine, at that specific point, and we can say that there are the nucleotide present. So a lot of nucleotides are present inside the nucleus, so it will be added here. Okay, and then if there is C, so it will bring G. If there is T, it will bring A. If there is A, it will bring U, and the process will be carry on, and the nucleotide added one in after other. But here one question arises in our mind, that how these nucleotides will be added in this process or in the machinery. In this case, as we talked, that we need this one transcriptional factor P, transcription elongation factor 2, transcription factor B. So this one transcription factor, elongation transcription factor, will be added here, and they will receive the nucleotide. The RNA polymerase gives signal to this transcriptional factor, and the job of this transcription factor is to bring or to receive the RNA nucleotide. So these are the RNA nucleotide. Okay, and then it will added RNA nucleotide one in after other. Now, how the RNA nucleotide are added one in after other? Let me a little bit explain this phenomenon. What happened? Remember, if we see the structure of nucleotide, so let's suppose this is not the exact structure, but let's suppose this is the nucleotide. Remember, they have also 5' end and 3' end. So there is the 5' end, and here is the 3' end. So at 5' end, there will be phosphate group, 3 phosphate group there will be. One is called alpha, second is called beta, and then gamma, I think. Okay, now what happened at the 3' end, there will be the hydroxyl group. Okay, so how the next nucleotide, RNA nucleotide, is added to the first RNA nucleotide? The addition is possible like that. Again, we have the nucleotide, as you can see. They have again a hydroxyl group. So we need this 3' hydroxyl group. With this 3' hydroxyl group, there is a lone pair, as you can see. These dot can represent the lone pair. These are the lone pair. This lone pair, these are the again 3 phosphate. Okay, this lone pair have the ability to attack the alpha phosphate, so it will attack the alpha phosphate. Now once it will attack the alpha phosphate, so these 2 phosphate will be released out in the form of pyrophosphate. So we get pyrophosphate, Pi. This Pi have energy, so the energy is stored between them, and when it will break down, as you can see, the bond is break down between them. So after that, it will release the ATP, which is the energy currency. So once it will release the ATP energy, so after that, energy will be used by this RNA polymerase, and it will be move or carry on. So we can say that how the RNA will be carry on? It will be carry on by this energy, which is obtained by the breakdown of this pyrophosphate. Now once the pyrophosphate are released out, so then we just have 1 phosphate and the hydroxyl group. So after that, the nucleotide will be added one in after other like that, man. So there will be phosphate group. So phosphodiester bond will be formed between each nucleotide, as you can see. And after that, another nucleotide will be added like that manner. Then 4th, 5th, 6th, 7th, and the addition of nucleotide will be carry on, and we get a long chain. So this is how the nucleotide will be added one in after other. Now after that, what happened? These 2 factors are recruited. And this is, let's suppose, the long chain of the RNA. Now at 5' end, almost 60 to 50 to 60. After the addition of 50 to 60 nucleotide, then what happened? 5' capping will be occur. So this is the 5' end. So here a capping will be formed. how the capping will be formed, what's going on during capping, what are the modification means how the 5 prime will be modified due to which the cap will be formed. We will talk it later. Now this is called 5 prime capping, but how it will be occur, we will talk it later. So how it will be formed? It will be formed after the 50 to 60 nucleotide addition. Okay, and it will be possible by these two factors, is spt5 and TAT-SUF. Okay, then let's this is all about the elongation of DNA DNA transcription in case of eukaryote. But remember, as we talked, that the RNA polymerase, in this case we talked that what are the main function of the RNA polymerase in prokaryotes. We talked that they are not perfect as DNA polymerase. We talked DNA polymerase is very perfect enzyme because they can add a correct nucleotide. But if we compare this RNA polymerase with DNA polymerase, so this RNA polymerase don't have the ability to add the correct nucleotide. It will make a lot of mistake in addition of the RNA nucleotide. So sometimes what happen, we have a lot of mistakes, not a lot of, but a little bit mistake will be done. So wrong nucleotide will be added. So we can say this RNA polymerase 2 is modified. Now this modified version of RNA polymerase 2 have the ability that they will scan, means they have also proofreading activity, and they will scan the nucleotide, this RNA sequences again, and once they found any wrong nucleotide, they will cleave out it and add the correct nucleotide. So this is called proofreading, and this is called exonuclease activity as well. So we can say they have also exonuclease activity, means they will cleave the wrong nucleotide and add the correct nucleotide, and they also scan the RNA chain. So this is all about the elongation of DNA transcription. I hope you guys get an idea about the elongation of DNA transcription. Inshallah, in the next video lecture, we are going to talk about the termination of DNA transcription in a detail. So please stay tuned and keep watching my video. Thank you so much.